physics, I don't know what fraction of this will become part of the
Maybe it's
complex,
only,
right?
yeah.
Exactly.
In case of looking at the reaction event, you have to track different cases. Eventually, one of the text actually goes together.
Yeah, exactly.
Yeah.
Yeah,
And then you discover that different text give you different complex businesses. And then you write another text. Exactly which we believe is part of the process. And then it's circumscribed like this.
yeah, after the series is also showing that the two one bigger and the other smaller. You do not really know what is the functions of that here, but I do know that there could be different forms of those applications of lenses. So there was a paper that came out from the elements where they did the microscopy. Standard paper.
Is that a paper about basically?
I think they're tomography.
Yeah, I know the paper.
Yeah, so exactly. So they do tomography and they makes a zipper, kind of to.
Well, they think they form this membrane associated thing, right?
Yeah, exactly. So they call it a zipper.
Yeah.
That connects those two ends with the endoplasmic reticulum.
Just barely.
Yeah, that's the table. But let's take it long.
Things like that.
A lot of wines are working flavors, yankees, yellow fever, anthrovirus, coronavirus, seasonal as well as.
Great. Oh, yeah.
So tomorrow we're having an in-house seminar with Dimitri.
Okay, yeah, this is really good that this will allow me I was just talking before about the growth
Yeah, well, this spring break is not very good.
NSP6 is challenging because it has 6 transfer lines. So we are just trying to take
Don't modify anything. So the memory is fine. You just have to do the detection.
But we still have to add a tag.
Oh, that, yeah. Yes. If you want to fish it out, you need combination.
It's easy to put tags in NSP because people have put tags in NSP like house coronaviruses. So I think we're going to stay the same approach. And I think people have put a tag.
Yeah, we have some experience of common protein, so we can share those things and we see whether this can be helpful.
Those small details, right? Yeah, exactly.
So we actually got it proteins.
Yeah, okay.
But they don't work because they don't come together.
Right. So basically,
we also try to separate.
But you did not have the complex.
Because 2 projects better, and then 4 and 6 individually.
Well, I have an interesting history there, right? Do you know my history? I'll tell you.
Yeah, a couple of minutes. All right, we can get started, and I can give really long introduction, or we can give a few more minutes, and then I can give a short introduction. But that's kind of a problem to give short introduction to Hong because there's just many things we can say about him. Well, first, of course, the standard information. Right? So, Dr. Zhou is a graduate of the University of Science and Technology in Hefei, China, where he received his bachelor's and master's degree. He then moved to Houston, Texas for his graduate studies, and then he stayed on in Houston as a postdoc and ultimately received a faculty position at the School of Medicine, University of Texas, Houston, where he originally worked on other things. He's a physicist by background, if anything, computational biologist, until his interest eventually crystallized on structural biology and ultimately cryo-EM, which is kind of the illustrious part of it that got a major boost when he moved to UCLA in 2007 and became a founding director of the cryo-EM Center at UCLA, California Institute for Nanomachines. So nanomachines is a theme where the scale is reflected, and in essence is reflected in the title. Right? And this is what he's been working on ever since. He received multiple awards, including Pew Award, the Established Investigator from American Heart Association, and he is currently at UCLA as professor of molecular microbiology, immunology, and molecular genetics. So it's this long department. We have been fortunate to collaborate with him on a number of years over some very challenging projects, and his visit to Boston is called, I think, perfect timing. So he was in time to run a marathon, and he was in time for BU, that is starting to venture into the cryo-EM field, to actually get some first-hand, invaluable advice on actually how to make cryo-EM enterprise run at the place where it wasn't before, right? So I think we can all benefit from this discussion. But after that rather long introduction, I'm happy to give you Zhou with his seminar entitled Captured Viral Genome Packaging and Transcription by Cryo-EM. Hong.
Let me get you set up with microphones.
Can I be heard in the back? Thank you so much. Well, thank you so much, Rosalie, for the kind introduction, and Nelson for the threads to come into genomics. So give me an opportunity to talk about genomes, which I thought cryo-EM can make a contribution. So I should start by mentioning that I was very great actually for me to catch up with pioneers in the field of cryo-EM. In the back, Chris, Aki, and Esther back there. We struggled together 30-some years ago when cryo-EM was being modeled by physiographers and others. Being that we can only have one bit of information content because we can only tell yes or no whether we have a sample or not. So, luckily for all of us, we survived the challenges over the years, and then we sustained our career in this field. So today I thought I'm going to take the opportunity to really tell you a story that I decided to do early on in my scientific career, to say that try to push cryo-EM in one area of biology that I think is sort of unique, that we offer some unique advantage compared to traditional structural biology field, namely looking at beyond proteins. So, so I decided to talk about viruses because those are good tools that structural biology always used to demonstrate technology, and in this case, it gives us a wonderful tool to look into the genome and how genome replication transcription occurs. So I'm going to talk about, for those of you who know cryo-EM is technology, just bear with me for the next maybe 5 minutes or so, because I really, by talking to you, Esther and Chris today, I know that you guys have good instrument being ordered. And this is a perfect time to actually take advantage of those to be set up locally. So just give you a little bit update about what is the key advancement in cryo-EM in the last 10 years, what sort of enabled that this become a tool of common utility. I think the two things that I was mentioning to keep in mind compared to the traditional EM, cryo-EM, that the two things are very important. One is that the what's called counting camera. So contain electrons. So because we used to record images 30 years ago on films, and then that required chemistry to develop on film to have a signal. And then about 10 years ago, that we began to have this technology that you can record electrons. Basically, electron camera, instead of a CCD camera you have your cell phone, you have the electron camera. Then you can record images directly without beyond the film chemistry. And then, in the meantime, computer is such a good thing. The computer speed increased about 1,000, maybe more times, based on the Moore's law, that enables the image can be recorded, not only recorded, but also saved into disk very rapidly. So we can record about, say, 400 images per second, so that you can basically record an image with a single electrons. So once you do that, then you can start counting electrons. And that really changed the field, because now you can tell that I have a single electron, that means you have a signal, or not. Well, not, it's noise, the background. So basically, that really touch upon the very fundamental limitation of crowd EM, namely that is radiation damage. But radiation damage, then to cope with that, we introduced crowd EM, and then and also try to reduce dosage to recording images. That reduction of dosage that basically make the crowd image traditionally very hard to see anything. Now, because of the present noise, because you can count electrons, so you can, in principle, you can get noise-free images. So every great image is a good image because you have signal. You may still not be able to see things well, but it's much better than the traditional crowd images. So this is a typical image recorded one of the first crowd images from electron detectors. You can see that. You can see these are viral particles frozen. So when you water, there's no stain. Then you can see proteins outside and those things inside. Those are nucleic acids. You can see clearly. So the signal is there, and then now, then you can do image processing and try to. The next thing is try to find the relationship among those particles. So we make the assumption in crowd EM. Those are sort of identical structurally. They're the same thing. Okay, and then they differ in their appearance because the orientation are different in solution. Then the task is to figure out the orientation. Then the computer scientists will tell you that you can do that. You can just try to compare them. You can figure out the orientation. Once you figure out the orientation of each of those particles, they're the same. They represent different views of the same structure. They can just put together by merging them together using the technology everybody's familiar with, because it basically is like CT scan. CT scan that you have images of your body recorded by rotating the camera around you. You know the orientation. Now you know the orientation of each object, and then you have different views. You can use the same technology put together in computers. You get a structure like this. In this case, instead of the CT scan, then these electrons, the wavelength is very short, so you can have high resolution. Then you have a three-dimensional view of your objects, and then if you accomplish the resolution about 3 angstrom resolution, which is we do normally in biology, then you can tell the building blocks of biology, namely amino acids and nucleotide bases. You can actually tell them, resolve them by 3 angstrom, and then you can then trace the chains of those proteins and nucleic acids and build atomic models by combining your sequence information. And that's what we do. So we do a structure like this, then you can build atomic model. So here, and then you do that not only protein, if you have nucleic acids, you can also resolve nucleic acids. So the key take home here is that because you don't actually need to know what you are looking at, right? So when you actually determine structure, you can trace the chain, you can actually tell what you have, and then you can identify. If you actually roughly know the sequence, you can actually build models. As shown here, this is a model that we did for one of the viruses. I'll tell you in a moment. This is inside the virus. They can see this. This is a Watson-Crick base pairing of the nucleic acids. So you can actually build atomic models of the genome. So another things I should mention here, this is actually a virus later, so you can actually visualize the modification, like John study glycosylation. Yeah, you can see those modifications in proteins. In fact, many people take advantage of those. They want to look at the native sample to discover those sugars, modification on proteins, or other things. So the bottom line is that I'm going to talk about today, talk about viruses, but you can do many different things. Of course, nowadays people do cellular biology. One of the things I try to promote with working together is that you take a sample from a cellular context, hopefully in a native form, and image that in a sufficient resolution so you can identify them, and then try to minimize the purification. Don't do purification. That's one thing I want to advise. You try to look at this like Chris Aki doing. Just take the membrane with the pore, image it, and then you can, if you look at different things, then you can identify them based on the structure. And then the surprise is, the good surprise of individually discovery is that you will discover things you never thought of, different states. In the case with Rusin's project, we discovered those different states in the additive processes, and which is very happy surprise to all of us when we saw that. So that's a unique difference of sort of biology enabled by cryo-EM. Namely, instead of doing this sort of top-down, you know at these ends, you actually just look at everything in the bottom and then discover what you have and try to interpret that what you have. So it's sort of, I go fishing, basically, which I enjoy typically. So you'll be happy to have the surprises. This is sort of an approach that we are pushing. Of course, if you do have your sample of interest, that is the things you have been working on for the last 20 or 30 years, you want to focus on that, that's fine. Yeah, you can work on that. And with the traditional crystallography approach, you have a protein imager into the structures. And that process typically will be easier because you probably have a well-focused problem, and then your sample probably well behaved. But if you typically, another thing we discover is that if you do that for big complexes, then they may not be very stable, and then cryo-EM may not be very cooperative if you do that. So the seasoned cryo-EM people in the audience can tell you about those, but that's beyond the topic of today. But nice, I'm going to talk about viruses today. So viruses are very easy to begin with because cryo-EM people start with viruses because a very simple reason, because they do have symmetry typically. Okay, the two kind of viruses, these spherical viruses, the helical viruses, similar to the Esther study on the spherical, but today's virus will be all spherical, the sphere. They typically have a part of the virus being symmetrical, namely the capsids. They show here. This is a herpes virus, and they have symmetry. You can see those highly symmetrical arrangements. These are typical icosahedral symmetry, and then, and that's the reason people started using viruses in the early days for technology development, because then that reduced the amount of work you have to do, because there's 60 copies of the same thing. So taking one image, actually currently doing 60 times, so that reduced the workload. So if you don't have a fast computer, those times, that's pretty good then. So what I'm going to talk about today is beyond protein. So the idea is for it. So for viruses, then you typically have one genome. Okay, in essence, there's only one molecule per particle. So obviously it doesn't have the symmetry. So what we do is that you sort of treat the virus, it's like a ribosome. Okay, but it's more complex than ribosome because that has symmetrical components, and then which is the protein components. Then you should, so we divide with a mechanism that can take advantage of that, make an assumption that the protein and genome are interacting specifically. So there's only one arrangement. Okay, so it's sort of, then there are 60 different locations of the virus that interact, so that possibly can arrange your genome. So you need to fish out that one. So this one out of 60 arrangement, you fish out. There's one way to get it. You can take advantage of the symmetry. You do this cyclically. The bottom line is that you take advantage of the symmetry and then try to fish out the possible arrangement based on computational classification. So the story is long, but I'm going to just tell you that there's a procedure to develop we call subparticle construction. which these methods can not only apply to virus like this, but also can apply for helical filaments. Some other things like cellular complexes have certain arrangement that then have symmetry, but then has asymmetric attachment. Then you can take advantage of those to fish out the asymmetrical attached components. So this is a common approach in cryo-EM nowadays. So the topic of virus, for those who actually do not look at virus on a daily basis, so I'm going to tell you a very simple point of view of viruses. So I have interest in viruses because they're the simplest form of replication, right? They can replicate itself, but some have from cells, right? And then I thought this was interesting to actually figure it out how it does that. So I look at a virus typically is two things. There's a information carrier, which is the nucleic acids, and there's a protein to protect it. So actually from the environment, as shown here. Then they need to somehow break out to enable the transcription and need some other help, typically for cellular help. So of course, in the RNA virus is simpler because it doesn't need to go to the nucleus. It does not DNA typically. Then they can stay outside the nucleus and then stay in the cytoplasm, and that's actually how this works. In fact, if you could provide it, you can provide a system, supplement a system with enzymes and substrates in solution. You can actually do this in a cell-free environment. And then you can capture those. So it's a perfect system. If you really want to actually study this, you can use this kind of RNA virus to capture some important processes, for example, transcription or replication of the nucleic acids. You can do that on the grid because you can do this on a cell-free environment. And that is one of the topics we're going to talk about today, how actually take advantage of this process of RNA virus to capture transcriptional events using the EM grid, multiple states. And of course, for DNA virus, it's a little bit more complicated because it doesn't need to go to the nuclei. It's shown here, this is a simple version of nucleus. And then go to the nuclear pore here, insert it into the DNA, insert it into the nuclei of the cell, and then where the replication of DNA occurs, and then that need to come out somehow, also perhaps in the nuclear core complex, and then protein synthesis occurs in the cytoplasm. Some of the protein need to transfer back into the nuclei, and then the protein shell is being assembled, and the DNA, viral DNA, need to be inserted into the pre-assembled capsids, and then this whole thing come out through a process they show here through the egress pathway. So I'm going to cover today a process occurred inside the nuclei using the herpes virus. It happened to be a DNA virus, double-stranded DNA virus, and also a couple of viruses, RNA viruses, where only occur inside the cytoplasm. Where I'm going to talk about two kind of RNA virus. One is a single-stranded, which is relatively very simple, and then the other one is a double-stranded RNA. Try to draw some comparison among those, and hopefully that some of the lessons learned through this process about biology. So the first topic is the herpes virus. Herpes virus is not foreign to any of you because I can guarantee that everybody in this room already had it in some time of your life, simply because you're not babies anymore. And then you probably got this virus through your mother or through your partners, your use, or through kissing each other, and then also other intimate events. So there are a total of 8 kinds of herpes virus. The one that you typically have that bothers you is the coastal virus. Happens on the tips of the lips, one section here, and the other one, which is some once you're focusing on this, that you should be watchful of those is basically VZV that cause shingles. Those are very common when you reach the age of 50 years old. I had that event about 10 years ago because aging. is painful. That's one thing I can tell you. Just if you can take this vaccine, fortunately, if you can take that, it's very painful if you have that. And then, of course, this cytomegalovirus, which is actually a very medically important virus that causes eye infection, retinitis in the eye, and then also birth defects. It's a leading cause of birth defects in this country. And of course, the latest member of this virus is called compulsive sarcoma associated herpes virus. These are discovered actually in Los Angeles in the 80s because there's a huge population of AIDS patients, and then they thought this is, when they discovered the AIDS virus, they thought before that they thought it's infectious, and then because these people always have it, but it turned out it's another virus other than the HIV, and this is the latest discovered herpes virus, PSHV. Basically cause tumors, on skin tumors on those patients and other patients in Africa. The topic I'm going to cover today will be HSV and HCMV. This actually, they are all herpes virus, but there's distinction. And this on top, it's just vis-a-vis. You can see they occur on only one side of the body, right? Always on one side and here on one side of the body. And that means they are neurological. So it's they transfer through neurons inside the body. As you know, neurons are only inside it. That's why you only see it on one side of your body. On the other hand, the CMV this thing is not a neurological virus, but it has a very large DNA compared to other herpes. It's about 50% bigger than those other herpes viruses. Since we're talking about DNA related stuff, those are used as a comparison. So the first structure I'm going to share with you is actually this is my thesis project. Okay, I'm an old person, so that means I failed miserably as a student. When I was a student physics major, I didn't know anything about biology, but there's a guy called Zhou, medical medicine. He failed to retain students, biological students in medical medicine, and then he decided during that time that he's not going to recruit any student who actually know biology, knew biology, knew it. So that was me. So I didn't know anything about recruiting by Zhou to study. He wanted to develop methods called cryo-EM at that time. I wanted to study biology. I have no opportunity. I mean, here's this is a spot for me, so I ran for the opportunity. But while giving me this is a project called the herpes virus, because it's medically important. And then the long story short is that I did not accomplish what asked me to do. I did manage to graduate, to show that you could not improve the resolution, but we could not interpret the structure. We did the structure about 10 instrument resolution. So we see things, they see the sausage-looking things, but we don't know how to interpret. Okay, for obvious reasons, because we don't know the protein identity. Because this virus, it contains many proteins. It contains the genome contains more than one. It codes for more than 100 genes, and then we roughly know about 7 of those actually go to the capsids, and that's the subject. The bottom line is that after many years after I graduated, and then I've been watching what Zhou's been doing. There's nobody working on viruses anymore in Israel. So one of these days, about 10 years ago, when I was in a meeting with Zhou, and then asked so who is actually doing my thesis work? I didn't see any papers. And then Zhou told me I could not get anybody who is willing to do it. I give up on that. And it turned out actually he tried to recruit a couple of people, postdocs, to do it. They all quit on him. And then he said, well, I'm too old. Why don't you finish your project? Okay, that was the story. That's a true story. You can come with Zhou, okay? Yeah, we give him permission to work on this after 10 years. And then so my student, Xinhong Dai, who is a professor now in Case Western, he finished the project with me. So these are two other papers we did together, basically, Xinhong and me. The bottom line is that we did a structure like this. This is the structure shown in the middle. This is the herpes virus capsids. Okay, it's determined about 3.5 instrument resolution. Okay, you can see the image, of course. This is the pentamers, hexamers, the things in the middle, they join these things, we call triplets because they link with this. Okay. So once you, as I said, once you determine the structure about 3.4 three-dimensional resolution, the benefit is that actually you can identify this. And then we're able to interpret this now. So the most interesting discovery of this work is the following. So if there's multiple proteins, the most important protein is called major capsid protein, MCP. Okay. In the MCP, there are two versions of that. There's a hexagon version, there's a pentagon version here, and they're actually made of the same thing, but actually with different structures. So you're able to determine the structure of these. They look very similar in the upper domain, but in the shell, okay, capsid shell domain, they're very different. The same sequence of 48 different structures. This is a very common thing in viruses because the quasi-equivalence. Basically, the chemically unique positions, they have to adapt to the environment. So the interesting discovery of this, if you look at this protein structure of the MCP, then this is the sort of plotting against all the of this structure, looking at it differently, looking at the secondary structure composition and domains. So this is the upper domain, which is this big portion, and this is what we call the capsid shell region, okay, and which I call Johnson fold, because Jack Johnson discovered this. Photographer, when he studied herpes, studied GP5 of phage HK97. So this is a typical structure that been discovered about 10 years ago at that time in the phage. You can look at the secondary structure arrangement of these. They're very similar. See that the arrangement is all similar, except that all these loops got truncated and then insert a big domain. Each of those. So they're making a protein that is 3 times bigger than the phage itself, the phage protein. And then the insertion occurs not only on the upper outside, but also inside of the virus, and also the 2 termini of the protein shown here. So this is one of the next big things to learn, is that for many DNA viruses, this is actually a common thing. So no matter what you do, the DNA virus, this is one of the most common strategy to have a protein. So if you study phages and DNA viruses, they're almost to have this structure. And then this fold actually recently discovered in many cellular complexes also. So what we're thinking is that this is actually a very good fold that is unique to build large things that are very stable. Because virus DNA is a very double-stranded DNA doesn't like to be bent. So the only contain this DNA is to make a genome, and you need a lot of force to contain this thing. So this apparently is a very good strategy to contain the very stable structures to contain the DNA. And then this very stable structure is stabilized further from both outside and inside. From outside, there's a protein called small capsid protein SCP. From inside, also these other proteins, for example, here, these are triplex protein. Triplex here is a heterotrimer of 2 proteins as shown here. So this fold, these other proteins between these, also they're sort of, you can find similar folds in some, not all, phages. For example, this phage lambda GPD has a similar architecture of these proteins. Very small version of that, and then in the case of lambda is a homotrimer. In the case of herpes, it's a heterotrimer with different structures. So, so beyond that, sort of capsid structures, there are other things. For example, here, so if you look carefully here on the pentamers, the proteins decorated in the pentons, which are shown here, those are the proteins which are called tegument proteins. And those are unique to the herpes, the human virus. They're not in any of the phages. And those are the proteins perhaps are related to how this virus evolved to, to interact with different kind of cells, the tropism, different cells. And in fact, if you compare different herpes viruses, actually we study almost all the herpes viruses mentioned, and those structures are determined subsequently. If you compare those structures, the capsid structure is very similar, and then it is this additional layer of tegument proteins that are different across different herpes viruses. And in this particular case, this protein had this coil helices And then that's involved in these are the so put together in that protein. This is the main MCP, and this is a trimers. You see this additional protein right here. Those are the tegument protein. So if you look carefully, they have this color topology, and then of course when you look at this, they share structural sort of architectural similarity to many of the motor proteins, cellular motor proteins. So it makes sense. I mentioned that these are neuronal viruses. They need to go from here, of the source of infection, all the way to your brain, the central nervous system, and those involve typical long-distance travel, and those are transport is very efficient. So the virus interact with the cellular microtubule, there's a cytoskeleton, cellular highways to enable this transport. So basically, those are the protein structures that we were able to discover through this process. So meantime, I mentioned that our main interest actually looking inside in the virus. So when you look inside, so if you look inside, then you actually look at the densities of this DNA. So we're able to identify densities that is being packaged. We do a unique position here, which is called portal, DNA translocation portal. So this one vortex that is unique. There's only one per virus, and then there we can see this density red. And you can see this in more detail, in atomic detail. Then this we interpret that being the last portion of the double-stranded DNA, sort of being retained after being inserted into the virus, and then right before maturation. So we're able to capture those. So and then the DNA is capped on top of here, and then this provides a mechanism for the capsid to hold this very unhappy, sort of spring-loaded DNA, double-stranded DNA inside the capsids. And then, so the way we look at this virus is that these are spring-loaded things. They have energy. They have stored energy. So when the right environment is available, for example, if you encounter the nuclear pore, okay, Chris, that's your stuff, right? The nuclear pore, we need to figure it out, but it does attach the nuclear pore complex, and then that attachment, the theory says that enable the sort of a control detachable of this cap that enable the spring to be ejected, shooting into the nucleus of the infected cell. Of course, that process needs to be investigated, and one of the things we are trying to do nowadays is using another variation of electron microscopy called electron tomography to look at the cellular infected cell, try to capture that process. So we don't have time to go through, but we also study different versions of those particles. So we're able to identify different stages of this portal. They have actually there. You can also see this portal complex prior to the DNA insertion, and then the non-story short is that their structure is very different. Okay, so by comparing those different states of viral packaging, then we can learn the process of this DNA insertion and also hopefully DNA injection in the long term. So, and then if you look at this more carefully in the details, so actually we can actually begin to trace the entire genome. So what we discover is that we see these are organized it's like a spoon organized, and then we're able to, because the double-stranded DNA, they're actually big. They're 20 angstroms in diameter, so you don't need a high resolution to be able to trace this. So we're able to trace a total of 7 layers of this DNA organized in this way, and then come up with a model of how this is organized. The interesting thing is that, so if you look at this, you can see the handedness of the DNA organization. So you can see the left-handed supercoiled DNA inside the virus. So this offer also offer a mechanism how you can condense the DNA. Because DNA charge is not very heavy, there's no histones involved in this process, and then how actually you can coil this together in such a spring-loaded loaded form. So another surprise discovery is when we look at this, we have a very interesting surprise. This is actually cytomegalovirus. It's human cytomegalovirus. So I already mentioned that this herpes virus differs from the cold sore herpes virus in that the DNA is much bigger, 50% bigger. So if you actually look at the DNA, they actually indeed much more entirely packaged. If you look at the DNA here, you see that DNA even twisted, which is a very unusual situation. Basically, fill every available possible space with there. And then we also discovered that on top of the hexons, instead of seeing things I just show you, then there's another density which is showing like blue here. First of all, the yellow. The yellow is a protein that is unique to cytomegalovirus. And then it's called PP150, phosphoprotein 150. So that protein is there because to stabilize these capsids, these are much more highly pressurized compared to herpes simplex virus. In addition, we saw this blue density which we could not interpret for a long time. And then we did some sequence analysis, the deep sequence on RNA, and we discovered this to be a human tRNA. So the RNA is a human source. So this is a DNA virus. Actually contain RNA. And the RNA is not a viral, it's a human source. And they are sort of stuck there on top of the yellow, orange color PP150 viral protein. And we believe this is further stabilized the capsids with DNA. We actually don't know whether that is true. So we are trying to figure out what that is due. Apparently tRNA is we think it's not only doing the transfer of as tRNA does, but also have probably some regulatory function, which we don't know whether that is the case in this CRV. We are trying to figure it out. So basically we discovered something because we are approach of go fishing, but we don't know what that fish does in this case. So that led us to sort of give our motivation to design further experiment to test this out. In addition to the other things we talk about here, the CMV, you can see this structure on top of one of the oral, which I mentioned already in the HSV case. And this is much more complex protein. So there's many more things on top of that tegment protein I talked to you about in the HSV, which is involved in the transport of capsids in the neuron. But this has much more things. It turns out there are things that involve in other process of the HCM infection. And then this is a work that is not published yet. That we are trying to organize this in the paper.
Hey, Ham?
Yes.
Is it every possible species of tRNA mixed together on the surface, or is it just one?
Yeah,
species?
in this case, we actually identified one. They say so we did a sequence of the RNA. So there's one particular RNA. I forgot which one.
So there's only one species.
Oh, yeah, that's much higher percentage than others. So it's not a unique one, but one is sticking out. So we actually don't know what the function what is the function of that. So this is that region of the complex that I show you. Just want to show you the complexity of this kind of structure in a virus. And then imagine that if you actually reconstitute this in the traditional way, then you won't be able to do that because we don't even know the composition. In this case, we can just take this approach and try to discover what is the relative situation. So obviously you notice that the structure actually have different resolution. And this is much higher resolution here, but they get lower and lower on the upper region. So one thing you could do nowadays with this kind of approach is that you can take advantage of the everyone know the AlphaFold prediction. So you can take advantage of those to interpret the structure that is not as good resolution as the other areas. So this is the case in regions of the top region that we want to to model this together. Okay, so moving on. So when this is done, we decided maybe we should get back to some of the other genomes. So the first one we decided to do is this virus called a phage. It's a single-stranded RNA phage, bacterial phage called MS2. Many of us probably use this as a tool on your whatever your favorite system of molecular biology. Of course, it turned out this is the first ever sequence organism because it's very simple. It only has about 3,500 bases, single-stranded RNA. It's two proteins only, capsids. There's a protein that make up the shell. There's a crystal structure early on published in 1990, and then there's always a mystery because this second structure is called maturation protein, MP, and that protein is part of the virus, but in the crystal structure, because the symmetry was imposed, and then we don't see that. Of course, the genome is not resolved in the crystal structure because the symmetrization in the structures. So we thought maybe this is a good sample to test the idea of regionalizing the genome inside. So this phage actually have a unique lifestyle because it's also because they involve in they sort of hijack the bacterial conjugation process. Basically, they attach to the pili of bacteria, E. coli, and then you can see that this attachment, these tomography images of those kind of viral phage attached to the pili. The idea is that they sort of enter the bacteria through this process, not through the traditional kind of way of fusion, like SARS-CoV-2. You have this chromatic protein here. It doesn't need any of those. Just somehow hijack this process of conjugation and enter the host. And it was a mystery. How can the virus do that, right? So we determine the structure is using sort of similar approach that I just told you, the herpes virus. And then there's two sort of very immediate observations we made. The first one, if you look at the outside, you see this unique region which are colored that is different from the other region, from the orange color. This is the we are then immediately identified as a maturation protein. So the other protein, of course, we already know from crystallography. So this is that mysterious missing protein that we are able to now resolve. The second thing is if you open up inside, you'll see this blue density. You can see inside, and then it's immediately operates when you look at this. There's the Watson Crick base pairing because you have the major groups and the minor groups of the Watson Crick base pairing kind of arrangement. So you say, how come? Because this is a single-stranded RNA. How can you see this thing? So it turn out, these are particularly near the region of the protein. They are actually pretty high resolution. For example, you can actually resolve the nuclear bases in those regions, and then so we are able to tell this actually sort of twisted single-stranded RNA, and then they sort of foreign these sort of stem groups, which is typical RNA structures of single-stranded RNA. So we are able to actually take advantage of those features, and then we are able to identify the structures on this side. So showing here is the same structure, just filtered. So you can see that this region, that's the MP region. You can see that they're different. So that's that MP protein. If you open them up, and then you'll see the inside structure inside. These are the genome structure inside. When you see that, not only see the base pairing major amino groups, you can see the branches of RNA density, and this turned out to be very useful information that we can use to sort of come up with a model of the genome organization. Because those are regions that make a contact and interact. So this because this phage being used as a tool in molecular biology, so we already know a lot in the literature that which region of the nucleus interacts with which. So that means we interpret this branch regions of interaction. So that enables us to come up with a model. So it's shown here, these are secondary structural prediction of the genome. So you can identify those regions of intact from the literature, and also the regions of stem loops from the prediction, and that enable us to construct a sort of preliminary model of the genome organization of this. So we are able to model about 80% of the sequence of the genomic sequence is shown here. For the most part, it's only a tracing of the backbone, but for regions attached to the protein, those regions are more sort of stabilized, so we're able to resolve them in a higher resolution. So we build a atomic model of the stem loops region shown here, and particularly for regions that interact with the MP protein. So you can see that the massive interaction with the MP maturation protein. The protein is called maturation because in the absence of this protein, you can also actually have a particle, but they actually could not be infectious. So people don't understand this. Now it seems to make sense because they need that to have the genome association. And then what happens is that really this association is very important because during the conjugation and the pili retracted product process, what we think is actually this virus attached to the F pillars shown here through the MP, and then that attachment is very strong. And then because MP is sort of attached to the genome, and this viral particle is too big to enter through that hole, and then all these are stripped off during that process. The sort of MP plus the genome sort of poured in through this process of conjugation. That's how the genomic material is sort of delivered into the host cell. In this case, it's a bacteria. The message that you learn from this is that the virus, they're not the same kind of virus. The virus, they're not built the same. They have all different kind of mechanisms of surviving and replication. I mean, it makes sense in biology, right? Whatever it works, they stay, right? That's the principle of Darwinism, right? So this is another example. So we sort of hijack this very unusual process of bacterial conjugation, and then enable the transfer of the genomic material into the host, where the host replicate the genome, and then you can then have the virus produced. So the last example I'm going to talk to you about in maybe another in the next 8 to 10 minutes is things that eventually used to actually develop a career for young focusing audience. This is my story. Okay, so about 25 years ago, I was on the job market. So then I got my job, and then I had to make a very critical decision. So what I decided to do to get my funding to sustain my research. We have a startup package as usual, but that only lasts for a very short time, right? So what I do to demonstrate you can do things, so people can retain you to move on. So as usual, it's advised by people, by old folks, and then you pick on something medically important, which is good advice. Right? Medically important, get advantage funding makes sense. So what I picked is following this advice is a virus called dengue virus. Wasn't probably low. Actually, I need to work on that. But it turned out I never got any funding out of it. So it's a failure also in that respect, because I never get any funding. As of today, I never got any funding out of the topic I chose. The dengue virus, 25 years ago, we know it's a very, very good part because it's very medically important, obviously, because it's medical at that time. But one thing I sort of missed, lessons learned, is that I haven't done enough homework to see who are the players in the field. So in my case, I only realized when I started seeing papers from Michael Rossman, because the survivors know who Michael is, and then your neighbor is Stephen Harrison, Harvard Medical School. They start chopping papers in Cell, Nature, and Science, and then realized I actually have a big competition. So the long story short is that every single time I have a structure, the paper come first, so I got scoop every single time. Eventually, I learned the lesson. I'm not going to work on any medically important virus anymore. I'm going to touch something that nobody wants to touch on, which is this guy called CPV. cytomegalovirus, so cytoplasmic CPD, is a virus that is double-stranded RNA virus. It's a member of the real variety, but it's a simple member because it only have a single shell. Single shell, it contains a double-stranded RNA segments, in this case 10 segments. I picked this because it's an insect virus. That means it's not medically important. I figured there's nobody work on this. In fact, if you look at the literature, 10 years prior, there's not a single paper. So I'm making sure that nobody work on it. And then this actually in Japan, in Asian countries, like Japan and China, where rice is the main, nutrient source, and then people use this, they spread this on their rice crop and vegetable crops because there's an insect, there's a pesticide, the green pesticide, perfect, it's biological. So, and then the motivation for me is I can use this to study basic biology. But in this case, it's a double-stranded RNA virus, so it has all these enzymes that is retained for the virus because double-stranded RNA is foreign to the cell. Right, it's foreign, so you cannot actually have a double-stranded RNA inside the cell because it's foreign. So the virus retain all these enzymes to do the capping and do the transcription inside the virus. So I figured this is a perfect tool because then you can do this thing in a cell-free environment. Then you can actually capture this process per crowd again. So I'm going to tell you the story that what we did using this virus. So it turned out, of course, historically, this virus actually being, I recognize this virus because these guys actually, the people published in the 70s. And I was picking up this in the 90s, so 20 years. So this virus was used to discover the capping process, focusing old enough, you remember those things we learned from chemistry textbook. This is how the ion capping is discovered using this virus. Again, it's very simple, it's very easy to handle, you don't need to deal with safety protocol stuff. So we take this, in this case, we can take the virus and add all the dense nutrients, and then in the buffer, then you can actually trigger the process of transcription in a cell-free environment, and then this is the image. So you can do that image, you can see those RNA being synthesized and released, and hopefully it's already capped, so we can actually capture this process. That's the idea behind this. So we did all that, so just show that over the years we published a number of papers. So this is a paper that we document that you can see this dense sticky knot. There's a traumatic spikes. This actually is non-angular virus, but actually contain spikes that are very sort of reminiscent to what you have been used to see, like influenza virus spikes, SARS-CoV-2. They're very similar domains. They found the traumatic. They have multiple conformations, and then those are the spikes involved in the process of attachment. There's no fusion, but there's attachment. Then this virus enter not by through fusion, but through punching the hole on the inner zones. So the bottom line is, if you look at those inside, you can begin to see the blue density inside here, sort of surrounding this light blue, which are the polymerase density. So these are very characteristic because it has this very extended. Because again, double-stranded nucleus doesn't like to be bended. They stay sort of very long unbended state. So if you look at this carefully, breaking up, you can see that the total of 10 regions that you have this density. That's why I need us to interpret this being the polymerase inside the virus, and then 10 segments, the 10 enzyme complex. So that the reason that each segment have one is unique own polymerase complex to do the transcription for the RNA. So the idea is to take advantage of this ability to visualize the RNA inside. Here, just to convince you, those are actually you can do high resolution if you focus this region, and these are the region because what we call subparticle construction in the high resolution. This is the RNA-dependent RNA polymerase. It's actually this complex contains 2 proteins, HBP4 plus the polymerase itself. From this, it's sitting on top of the capsids. So interacting with the, if you look at low resolution, you can see the again the Watson Crick base barrier. You can see the major groups and many groups of those double-stranded RNA. The other is the polymerase complex, and then we're able to visualize the structures that is sort of reminiscent to a structure determined by Harrison, because the I mean, not far away from here. In the case of crystallography, they can actually express this. You can see those structures. So now these are being captured in situ on the virus, and then we show that you can actually do this even in action. So you can actually capture those during the polymerization of the RNA. So we do that by adding all these other required elements in the NTP space. Of course, HCU need to be added if you want to have a continuous transcription. But we can take advantage of these. We can just take any of those, just only 3, maybe 2, and based on the sequence of the RNA, then you can actually sort of capture sort of intermediate states, because you cannot proceed. You actually can proceed, and you can capture those states. So if you add all of them, and then you see a transcribing particle, so you can see there's a reaction. So basically, we did all this experiment, many structures being captured by doing this, and then we were able to sort it out. These are the states. These are different kind of structure we are able to capture by doing this process. So basically, we can classify into 5 categories. One is quiescent, there's nothing going on yet. And then there's an initiation, when you add the SAM that triggers the action. So you sort of start it, but there's nothing to continue because there's no substrate. And then also this elongation step where all the sort of RNA, the NTPs provided. And then also this state which is sort of characterized as a body of state. These things are not going well. And then this early elongation, if you only added limited only 3 kind of the nucleotide, for example. And then so this is the RNA capture. You can see that this is double stranded, and this is only this is the cap is actually binding to the side of the polymerase, so cap binding site. And here is during initiation, this the double stranded RNA is sort of branch out. So you can see that this one branch is enter into the hole there, and we interpret that being a template strand, and one stay outside, the non-template strand. And then during the elongation, you can see that this is transcription bubble being visualized. And then interestingly, we visualize for the first time ever for any transcription reaction that the non-transcript strand and the template strand sort of reunited with each other. This is a genomic RNA, and this is the lacing messenger RNA sort of being released, being captured also. So apparently, but from this structure, you can see that the way the RNA sort of know where to go is because of the charge property, which I'll show you in a moment. And then these other states, they somehow the polymerase detected is not productive. They sort of retract and try to start over again. So we're able to capture those states. So just to show that we're able to actually resolve those details in the reaction center. Here, we able to model this. We also capture some ATP inside in the reaction center. And this is a messenger RNA. Just to show you that this, you can actually see this in detail. So again, this is the capsid. This is the polymerase complex with the transcription bubble, and then the lacing messenger RNA being released. So you can see that these are relatively low resolution. Regions like here is much better resolution. So we can actually model this atomically in this region with the density, which you can see in a moment. You can see the high resolution structure here being modeled. So we're able to build an atomic model for this part of the catalysis. And then the interesting model we came up with is that, so because we are able to capture those states, then we think as a physicist, we think this is sort of energetically. So what we think is that in a sort of quiescent state, they're not the most stable states in our opinion. They're sort of metastable state. So they can stay there if you sort of don't have any nutrients provided. In this case, you need SAM, which somehow triggered the reaction to occur. and then add ATP. Then that provide energy to sort of jump through this transitional state, and then the initiation state is probably lowest energy state that you can keep producing. And then you provide the NTPs, because NTP is being consumed, this provide the energy source, and then this can continue as shown here. Of course, the body elongation state, they're probably in the sort of middle, they can actually track back and start over again. So this also explains that in this model, also explains how this virus can be a nanomachine. Basically, they can be produced, they can generate this RNA continuously, and then so it was always a puzzle. So in this model, so the polymerase was be stationary, attached to the capsid, and then some of this RNA, the genomic RNA, they'll start transcribing as shown here, and then this is the tail of that, and then during the transcription, and this tail sort of moving around, and then sort of the leading template region will chase this tail during this transcription cycle. So we call this model we call Ouroboros. Those actually with Greek mystery. This is the model. Basically, the snake sort of chasing the cell, chasing the tail. So you can see that you can actually kind of reproduce the messaging RNA this way continuously. So I hope I told you today that probably is a wonderful tool. You can actually study going beyond protein. In this case, we show you that we use a virus. You can do these genome structures of viruses. Show the herpes virus. The most interesting discovery that we made actually the MCP actually very similar to phage. It just made it more complicated by adding those different domains, like the regions of groups, and then you have the portal. You can capture the DNA, retention state, and then I show you the CRNV. We're able to capture the host RNA, and also the phage, single-strand RNA phage, MS2. We're able to resolve the genome inside. And in the case of the double-stranded RNA, I'll show you that we use that as a tool to capture this intermediate states of transcription. So with that, I'm going to thank people who actually did the work. Mainly Wintal, who did it together with Jonathan G. There's a postdoc and graduate student in the lab working on the herpes virus together with David Rogaski, discovering the tRNA. Other main players, those are already left the lab, Radio, Elon Zhang, those are the people who are involved in the single-stranded and double-stranded RNA virus work. And the work is in collaboration with members of our UCL community, ransom, herpes virus, and also in the CPV, agriculture, South China Agricultural University, James and Song, who work on the CPV is pesticide. So with that, thank you so much for your attention.
Questions? Tough competition. Millisecond, Nelson bits.
Oh, that was a fantastic talk. 2 questions. Can you treat CMV with RNase to affect its infectivity if you have all these tRNAs on the surface? And second question, can the EM see cations in your structure that mediate the RNA folding?
Yeah, the first question, no, because actually we want to show this actually integument layer. There's an additional layer of the CMV. It's an envelope virus, which I did not show today. So we remove those things in that structure. So if you want to keep that infectious, you need to retain those. So second question, cations, in the high resolution, typically you need about 2 angstrom resolution, then you can begin to interpret cation stuff. I mean, if you push even further, you can resolve protons. People talk about polymer. I mean, the best resolution you can do nowadays in cloud EM is about 1 angstrom resolution. Basically, if you want to see, ions, water molecules, then you need to push resolution further. I don't recommend to do that for viruses. They're too big. Pushing higher resolution might be more difficult. So typically, when you want to really resolve those details, then you do your sort of isolated system, perhaps a single protein, and then you push resolution.
Fascinating, John. Thank you. I actually have a question regarding the portal complex of the herpes viruses. So these complexes have so many different functions, like recognizing the package signals and then cleaving the concatenars and so on and so on. So what are we looking exactly here? Is it quiescent, like in the virion package? And then if that is the case, is it possible to actually capture these different steps? So how the DNA is reeled in and how the endonuclease cleaves and so on and so on?
Yeah, well, fantastic question. So, so double-stranded DNA virus like herpes virus, the packaging of DNA involves the cleavage of DNA because their DNA is much longer. And then, so what I showed you here today, what is specifically is actually the virion. So it's the already mature virus. So obviously we don't capture those intermediate states. So we have been made another effort to try to capture those intermediate states. What we do is actually we try to isolate those intermediate states. So in the case of herpes virus, the terminates that are cutting the DNA is actually temporary association. So they're not being packaged in the virion. So you don't want to see this in the virion. So what we do, we actually try to introduce some mutation to the terminates and try to somehow stop the process of maturation. So far, our effort has not been successful yet. But the protease itself has been actually expressed, and then there's a controversy right now about whether they're actually in the working protease, whether it's pentamer and hexamer. People don't know yet, but the structure determined by the crystallography, by cryo EM of the recombinant terminus is actually hexamer. Okay.
Other questions?
So, when you're trying to package it, there has to be something pushing to create the pressure that then when
Right.
spring loads.
Right. So it's the terminase. So terminase actually ATP. In this case, is actually 3 proteins involved. There's a protein. Part of the 3 is actually is ATPase. So you actually take energy to insert the DNA. So that is the ATP is being consumed. That is being, of course, converted to the spring energy during the process.
And the part that's disordered in the center, is that the part that goes in last or first?
I think it's actually the last piece of the DNA. It's been that way simply because it's they're not restrained as much as the protein reactive ones. So we think because like anything, you need something that you need to anchor, right? You need to anchor. So I think the first part of the DNA actually found a ring anchor. Actually, I did not mention that, but it's the top of the ring, and I believe that's the first part of the DNA being inserted. So basically, suppose I'm the part of that, so DNA got inserted, and then you actually anchor there, and then they keep pushing, and then this supercoil if you do that.
So do people know whether it's sort of a stop-start thing?
Yeah. There's a sensor mechanism to actually tell the virus when it's actually going to stop doing that. We actually talk about that in the paper, right? We have actually identified some mechanism that could enable that. Basically, I think it sends some pressure inside, and then that will be corresponding to the where that will transmit through the protein portal, and then the terminase on top of that to actually enable the cut. There's also some sequence specificity about where the cut.
Just some curiosity, the tRNA that functions as a structural component of the capsid, is it amino acylated?
Actually, we do not look at it. Because the resolution is not that high.
As you mentioned, there's a specific tRNA or Isaac acceptor. Is there a chance that it's an amino acid affinity that drives that particular tRNA in the capsule?
Yeah, we should look into that. Yeah.
In the cell, in the normal condition, most tRNA would be amino acylated, right? And that's the cell distress to start. Just very close to home. Yes.
Add one more.
Yeah.
So, when you did the structure of the virus attached to the epipilus, did you use the attachment site as a marker for the orientation?
We actually simply just fit the density. There's no particular. So you fit the density. Obviously, that's our interpretation. But later on, the other folks who work on similar things, they improved resolution of that. So I think that interpretation turned out to be correct. So we don't have any marker, just because it's symmetric.
But that that one protein would be the marker. I mean, if
that
that's
will
how
be
it
the-
attaches, then
yeah.
it has to attach.
Right.
Then you know how it.
Yeah. So use the density as a marker basically.
Okay.
Yeah.
All right. Well, if there are no more questions, let's thank our speaker one more time. And if there's still some more questions in a more informal atmosphere, we're going to hang around here for a few more minutes. And most welcome to